speak on my project. It's well known that the mRNA is consists of 3 parts. One is the 5' untranslated region, and coding region, and the 3' untranslated region. And all of these forms integral parts of a messenger RNA. But there is another mRNA. No, I should say another RNA, which is only the 3' part of the messenger RNA, and its function and expression are very much different from the conventional mRNA. And I would like to start with the story of our research on such an RNA. Our strategy is from Dr. Makoto Noda, a Japanese scientist. And he used a malignant cell line called DT. The DT, which is derived from mouse cells and has a rather round shape. We transfected this line with a normal human cDNA library and observed if any flat revertant cell colony appeared. Then we isolated the formed flat revertant and isolated the plasmids which was transfected into it. After purification of these plasmids, we repeated the transfection once again, and then we observed that some purified plasmids yielded again flat revertants. And we think there may be some genes or cDNAs which is able to suppress malignant phenotype in these cDNA, in these purified plasmids. And this is just the structure of the cDNA library we used to transfect malignant cells. And this is revertant colonies which appeared in the transfected cells. Note the revertant has rather flat phenotype, rather flat shape, compared with the original round shape malignant cells. And these are just some plasmids we purified from the revertant cell colonies. And these are two fragments of the vector, which is yielded by the Barmach-1 digestion. And we noted that in this plasmid there is a very small insert, which was rather 0.5 kilobases. We called this P14.6. And with Dr. Noda's consent, I take this plasmid back to China to continue our research. Then firstly, we tested if the flat revertant cells are really with the reduced malignant phenotype. And we use, among other tests, we used the nude mice tests. This is the normal control. These are all the tumors formed by the malignant cells. And these are from the revertant cells. So we showed that the tumor sites of revertant cells are significantly smaller than the original malignant cells. And then we sequenced the insert of the P14.6 plasmid. This is the results of the sequencing. By searching the cDNA library, it is found that the P14.6 insert is just the central part of the 3' untranslated region for NFIR6 gene. This NFIR6 also named CEBP beta. So we showed that the sequence, the 3' UTR of the CEBP beta, when transfected itself without any other components into the malignant cells, it can reduce the malignant phenotype of the original cancer cells. And since then, several other 3' UTRs are also formed to exert tumor suppressor effects when artificially introduced as independent RNAs into malignant cells. These are just a part of literature published in that period. For example, alpha tropomyosin 3' UTR and 3' UTR of ribonucleotide reductase R1 and I2 messenger RNA, 3' UTR of MEL18 gene, and 3' UTR of prohibiting gene. And these facts indicate, one, some 3' UTRs may be active in the cells independently, that is, isolated from their original mRNAs. And two, these 3' UTRs are active in regulation of cell growth, maintaining normal phenotype of cells, that is, the reduction of malignant phenotype. And three, independent 3' UTRs may be a novel type of regulators of cellular physiological activities. And then we started to try to find the interaction of 3' UTR and cell proteins, that is, to try to find the mechanism of its introducing this malignant cell type reduction. Firstly, we used RNase protection assay to purify the cellular RNA, cellular proteins which can bind or interact with our 3' UTR RNA by using isotope techniques. And here, this is the autoradiograph showing that in the revertant cells, there is a bind formed by the binding protein and the radioactive RNA. In that time, we called it BNF, but we were not able to isolate this protein. But we continued to try to find the sites in the 3' UTR which can bind interacting proteins. And I just say the results of experiments. The results show that the binding may be on the 3' end of the 3' UTR RNA. Yeah, this is the detailed experiments. That shows that the binding of the interacting protein with our 3' UTR RNA is occurred on this region. That is the 3' end of the RNA, of the 3' UTR RNA. And this specificity, the characteristic of this region were AU rich, many A and many U. So the above mentioned results showed that the site of interaction of the independent CEBP beta 3' UTR RNA includes an AU-rich segment near terminus. Then our next question is the tumor suppression of the independent CEB beta 3' UTR RNA only effective for a specific cell line, or is it generally effective? And to answer this question, we have investigated the effects of rRNA in a human hepatocarcinoma cell line, SMC7721. Then we used the clone and the plasmid, purified plasmid of P146 to transfect the SMMC7721. These are just part of the results. These are shapes of the original control and the revertant cells. And here it was shown that the transfecting is effective. Many integrated 3' UTR sequence were found in the transfected cell strains. And here is the RNA expressed from the transfected cells. Then Another question is the CBB beta, how does the CBB beta 3' UTRNA cause the cell mitosis delay, and delay to what extent? These are results by for example, comparison between original malignant cells and the transfected resultant, which is called clone 1. And here is also this result. Here we analyzed the resultant 7721 cell, that is clone 1, analyzed its transcriptome consistency. And the resultant clone 1 was chosen from the cDNA array analysis to find the genes which is upregulated or downregulated after transfection. And these are another series of experiments. The aim of those experiments were to find the effective parts of our 3' untranslated region RNA. We deleted the 5' end and the 3' end of this RNA, and then to transfect it to the malignant 7721 hepatoma cells to see if any effect can be found. And here is the result. I just simply describe it. These curves show that the cell growth cell growth of those cells which was transfected with the plasmid clones deleted the 3' end or 5' end. The growth was accelerated comparing with the clone 1, and this is the malignant control. And similarly, the neuromice tumors are bigger in the cells transfected with deleted 3' UTRs, comparing with the NO that is not, and that is transfected with the no deleted, and with the plasmids without deletion. And we compared the structure of the 3' UTR. We found that the 3' part of the UTR found this stem loop structure, and when we deleted the 3' part of this RNA, its stem loop structure destroyed. So this may be the cause of the reduction of tumor suppression. Yeah, this is just the redisplay of the slide just showed, and that means the two methods. One was the RNA binding, that is RNase protection. Another is deletion. The two results were in a good agreement. Then we show the cellular. We want to find any protein, to identify any protein which can interact with our independent CEV beta 3' UTRNA and then lead to tumor suppression. I just say the results of the experiments. And this is the course of the experiments. The results show that there is a protein. There is a protein named HUR, which is originally an RNA binding protein, and it trends to bind to the AU-rich region of RNA. But in our case, when the AU-rich RNA was deleted, the HUR binding is also reduced, decreased. Then the RNA, then the tumor suppression effect were also reduced. And in the slides just shown, we found that the CEV beta 3' UTRNA interacts with some cellular proteins to reduce to yield the tumor suppression effect. And here is another series of experiments showing the 3' RNA interaction with cellular keratin 18 are also related with tumor suppression. And these experiments showed that the cellular keratin protein and oncogene PKC epsilon in vivo can interact with our CEV beta 3' RNA and then resulted in significant tumor suppression. Yeah, and these are the course of the experiments, which I don't go in depth. These are also. Then, brief conclusions. One, the independently expressed CEV beta 3' UTRNA exerts tumor suppression effect in mouse and human malignant cells. The effect was caused by direct interactions between this RNA and many cellular functional proteins, especially HUR and PKC epsilon. So, our results are the first demonstration that an independent 3' UTRNA can have gene regulation function in trans as a novel type of non-coding RNAs. In 1910, a paper by Australian scientists was published, which showed that the distinct RNAs can be expressed from many genes, 3' untranslated regions, and this was in good agreement with our series of results. Then we found by using the bioinformatics, we showed the natural evidence of independent CB beta 3' UTR in human tissue.
Dr. Liu?
Yeah.
We're in overtime. You need to.
And this is from which organ?
Let's test it.
Yeah, please. Sorry. I should find. The 3' independent 3' UTR RNA are expressed in human mouse and fly as a common genome-wide phenomenon. It was found that the independent CBV beta 3' UTR RNA does exist in human testes and some other organ and tissues. Then now I'm trying to apply bioinformatics to further characterize and to find its functions and existence in another tissues. Over. This is over. Thanks.